Let's talk about proteins. This is our third out of four groups of organic macromolecules. And this is we've done carbohydrates, we've done lipids, and now we're doing proteins that we consume. And what proteins are a macromolecule that's made from long, unbranched chains of amino acids. So we had carbohydrates, which were made from glucose monomers, and they could be branched or unbranched. We had lipids that were made from fatty acids. And now we're having proteins that are being made from amino acids. And an amino acid, the word amino has to do with this group over here in green, which just means it contains nitrogen. It is attached to a carbon, and on the other side of a carbon, we're going to have a carboxyl group, and then we have a side chain. And the side chain structure determines which of the various amino acids it is, and as well as whether or not that amino acid is polar, if it's non-polar, if it has a positive change, or if it has a positive charge, or if it has a negative charge. And there are only 20 amino acids. You do not have to learn the names of the 20 amino acids. And when you look at them, there are 10 that are not polar and 5 that are polar. You don't have to memorize that. We have 3 that have positive charge and 2 that have a negative charge, which basically means that there are going to be 10 that are going to like water and 10 that are not going to like water at first glance. And these 20 amino acids are the exact same 20 amino acids in every single life form, whether you're a cockroach or a human, doesn't matter. Now, when we look at the protein shape, we have 4 different levels of protein shape. The most basic protein shape is what we call the primary shape, and that is merely the sequence of amino acids. So think of it forming a long string of yarn. It's just the sequence, okay? So maybe it is 5 of these in a row, followed by 1 of those, followed by 3 of these, followed by 1 of those. It doesn't matter what it is. Whatever that sequence is, that is the primary shape, okay? Realizing, of course, if we have polar stuff and we have charged stuff, some things may be attracted to other stuff versus if we have non-polar stuff, it's not going to be attracted to other stuff. So that is going to lead us towards the secondary shape, which is hydrogen bonding is going to come into play. And hydrogen bonding is going to give us 2 different shapes. It's going to give us pleating, and it's also going to give us swirling. So it gives us what are, and you don't have to learn them as alpha helices and beta strands, but basically you're going to get a swirl or a helix and or a pleat. And so here they're going to be shown as red swirls and then purple pleats in the structure. Okay, so that whole sequence now has gotten shorter because of the swirling and the pleating. But it's still one long strand, and we still have areas that are charged that are going to be attracted to each other. Okay. So what is going to happen now is we're going to have 3-dimensional folding due to those side chains interacting with each other. So those positive and negatives are going to attract to each other and form ionic bonds going on there. That gives us our tertiary structure. And then for our final protein shape, our quaternary structure, more than one amino acid chain will interact or form bonds with each other. So with just one strand of yarn, how we fold it, we can make baby booties, or we can make a hat out of it. but with multiple strands of yarn that can interact with each other. And we can make like the front of a sweater and the back of a sweater and the arms, and then they can all interact with each other and we can make a complete sweater. And that's how we go from the primary to secondary to tertiary, the quaternary structure of the protein shape. And that's how it looks like. So complicated when you're going to look at pictures of what proteins look like. And so that's why in this class, we're going to tend not to show you these pictures of what a protein looks like, because for our purposes, the details are not going to be that important. And where the details are important, we will point out the specific details that are important. So as you can see, we've got side chains, we've got charges, we've got helices, and we've got pleats, and all of those things structurally are going to affect functions, because remember, structure and function affect each other. And so when we look at functions of proteins, we've got some big broad categories that things that proteins can do. One thing they can do is they can just give structural support, like in our hair, in our nails. Keratin, that's a classic example of a protein that gives us structural support. We have proteins that give transport. For instance, hemoglobin carries oxygen throughout our blood to drop it off a cell. So here is a hemoglobin molecule. We've got millions of them inside every red blood cell in our body. We also have proteins that go through the plasma membrane of our cells that allow things to enter and exit our cells for transportation. We have proteins that are involved in our defense. You may have heard about antibodies. That's an example of a protein involved in defense. We have proteins involved in motion in our muscles. We have proteins known as actin and myosin. We have other proteins as well, but these are proteins you may have heard of previously. Some of our hormones are proteins, some of our hormones are non-proteins, but an example of a hormone that is a protein that you almost certainly have heard of is insulin. Okay? And enzymes, almost all of our enzymes are proteins. Enzymes allow reactions to occur faster if the reaction was going to occur anyway. They cannot make a chemical reaction occur that would not have occurred. I will be talking specifically about enzyme in a slide shortly. One thing I want to talk about proteins that you may have heard about is when proteins become denatured, and what that means is when a protein is exposed to an extreme environment. For instance, if it is heated, or if it is frozen, or if it's exposed to a strong acid or a base or strong ions, what happens is the normal shape of the protein is changed, it is disrupted. And as a result of that, that protein change of shape results in change of function. So that denatured protein has lost its biological activity, meaning it no longer functions. Example of this are a group of diseases which are known as prion diseases. You ever heard of mad cow disease? That's an example of one. So talking about enzymes, enzymes are typically made of a long chain of amino acids and then are folded together. And on one part of the enzyme, there is what we call the active site. And in the active site, there is a polymer that will fit exactly. So if the active site is round, then we have a round polymer. If it's a square, then we have a square polymer. If it's a triangle, then we have a triangular polymer. It's going to fit, okay, and form an enzyme substrate complex. And when it binds to it, what happens is the bonds in that substrate are weakened so that it will break into what are called the reaction products. So the substrate, that polymer, is broken into smaller pieces. What's that reaction called? I want to pull back that memory from previous videos. What's that called? Is this going to require energy or is it going to release energy? Is this going to require water or is this going to release water? If you don't know, stop the video now, go back and review those slides, and then come back and pick it up here. Okay. So when we're looking at enzymes, they usually end in ACE, A-S-E. So let me talk about a really common enzyme you may know about, lactase. And lactase is the enzyme that breaks down lactose. And as you recall, ose is a sugar. So lactose is the sugar broken down by the enzyme lactase. So here is our sugar lactase. You may not recall, it's a dimer. Okay, so it's going to fit into, what was this part called? The active binding site of the enzyme lactase. In this case, lactose is the substrate. It fits in there, forms, what's this whole thing called? Go back here, an enzyme substrate complex. Okay. The bonds between, right here, this bond is broken, and it's going to give us our reaction products, which in this case is two monomers, a glucose and a galactose, two sugar monomers. And then we have our enzyme lactase ready to work again. So what happens in your body if you don't have lactase? If you are lactose intolerant? Well, then if you consume lactose, lactose goes into your digestive system. When it goes into your small intestine, this dimer has what we call an osmotic pull. It's going to be pulling water into your small intestine. And that extra water is going to be going into your large intestine. So you're going to feel very bloated. You're going to be gurgling a lot because you've got a bunch of extra liquid. And when that lactose, the sugar, reaches the part of your large intestine that has bacteria in it, because your large intestine is loaded with bacteria, that bacteria thinks it's at a buffet in Las Vegas and starts munching on that sugar, and it's in seventh heaven, and it starts breaking those bonds and releasing the glucose and galactose. Only a side effect of that bacteria breaking down the lactose is that bacteria releases gas. And so now you've got a bunch of liquid with a bunch of gas in it, and you are going to be having those liquid farts. Which is not a good thing. Which is people who know they are severely lactose intolerant are not going to be eating those things. So here's the thing. Hard cheeses have a lot less lactose in them than soft cheeses. So if you're eating a really aged cheese, you're probably okay. If you're eating soft cheeses, goat cheese, you're going to have trouble. If you're eating ice creams where you can read the ingredients, you understand the words cream and salt, you're going to have problems. If you're reading ice cream and it's 20 lines of chemicals you don't understand, you're probably okay. Even if you are okay, as you age, you tend to lose your levels of lactase. So you tend to become more lactose intolerant in your 40s and 50s and 60s. So your dietary habits will tend to change. So what can you do then? You can use lactase drops and add it to your lactose foods, you can use lactose-free milks, or you can use non-dairy milks. In other words, you can do milk products from almond and oats and cashews and other substances. So if you want to know the prevalence of where people in the world have a higher incidence of lactose intolerance, look at the parts of the world that don't have a bunch of cheese in their food. United States, we've got a bunch of cheese. France, they've got 110 national cheeses. Does China have a bunch of food with cheese? No. Why? Because they've got a more than 90% population that doesn't have any lactase. That's why. When we look, Native American populations did not have any lactase. Aborigines in Australia don't have any lactase. African Americans from the south of Africa don't have much lactase. Mayan populations don't have much lactase. So look at your ancestors and you can figure out how lactose intolerant you may or may not be. And with that, I am done with this section. We come back, we only have one more video about macromolecules to go. So thank you so much, and I will see you shortly.